Hello, I wrote my third year project on the prevalence, incidence and temporality of morbidity occurrence in 52 thoroughbred foals in southern England during their first 26 weeks of life. Ooh, that's exciting. So why did we bother? We all know that morbidity has significant impacts on animal welfare, but it's very difficult to quantify the impact unless you know what morbidity is occurring, how often it occurs, and which animals it occurs in. We also know that morbidity has significant financial impacts, not just on the direct impact of cost of treatment and staff time required, but also there is potential evidence that it may cause a failure to race or a failure to achieve full athletic potential. So if you're losing money at sales or due to the mortality of the foal, that's quite a significant loss to the industry. So to do this, I used a data set from the Albumin foal microbiome development, which is a longitudinal study investigating the foal microbiome. I took a population of 52 thoroughbred foals from southern England and extracted all the data where they had recorded every health event. So Sophie and I, my lab partner, extracted all of the incidents of morbidity and then categorized them into one of 7 categories. We then performed some descriptive statistics to analyze the incidence, prevalence, and temporal patterns of morbidity. So what do we find? Nice table. So of the 52 foals, 40 had at least one morbidity occurrence over the first 26 weeks of their life. There were a total of 91 morbidity occurrences. The range of morbidity occurrences from no morbidity occurrences to 5, the median being 2. Non-congenital morbidity was much more common than congenital morbidity, with about a third to 2/3 split. And the type of morbidity varies significantly between congenital and non-congenital morbidity. So congenital morbidity we saw massively, limb deformity was by far and away the most common, whereas non-congenital morbidity, trauma, gastrointestinal disease and respiratory disease were far more common. So we also looked at the overall incidence of morbidity. So overall the incidence was 0.2 occurrences of morbidity per 4 month at risk. However, as you can see from the above chart, the incidence of disease or morbidity is much, much higher in the first month and then decreases over time during the study. There was also variability in the types of morbidity that were seen at different times of life. So much earlier in the study, in months 1 and 2, we saw very high gastrointestinal morbidity levels, whereas we went a bit further on in the study and we saw an increase in respiratory disease. Interestingly, there was a peak in months 1 and 3 with urogenital disease, for instance, and then things like trauma were consistent across the study period. So why does this matter? What does this show us? It shows us that foals get different illnesses at different times of their life, and it shows that they decrease over time. For instance, we had consistent trauma, whereas with gastrointestinal disease much higher in months 1 and 2. The reason for this probably is multifactorial. So to do with management, to do with what the foals are fed, where they're kept, etc. It may be to do with susceptibility as they age. So there might be different receptors in the gut earlier in life compared to later, which might result in them getting different diseases at different times of their life. Whereas horses, as we probably all know having worked with them, are very good at injuring themselves. So that's consistent across the foal's life. There's also a big argument for immunity being a very important part of morbidity. Interestingly, in this study, we saw no instance of failure of passive transfer or septicemia, which has previously been noted as a very important part of immune development in the foal. However, we can, because of that, say that passive transfer of immunity and that development isn't the only thing that allows foals to get ill. Because we still saw 91 occurrences of morbidity, and whilst all of those will not have been infectious, some of them will have done. One of the problems we did experience in this study is that we couldn't definitively define or diagnose each and every disease. So it's hard to say exactly how much was infectious versus non-infectious, but it is an interesting route of further investigation. Again, coming back to the lack of septicemia, we're not entirely sure why this was. Because it's previously been noted in papers about 20 years ago that it's a very, very common cause of death in foals, whereas in the study we experienced no death at all and no instance of septicemia or failure of passive transfer. This could have been to do with the management of the foals, so they were very well managed in studs in southern England. It could have also been to do with the parent study. So the Albumin well foal study, or the microbiome development study, aims to study foals for the first 3 years of their lives. It's very difficult to study foals that are going to imminently die if you want to study them for 3 years. So this may have excluded some animals that were more likely to die from the study as a whole. So in the future, again, the Aurora Waterfall study is aiming to examine the events and the effect of microbiome health events and illnesses and athletic performance in 1-year-olds, 2-year-olds, 3-year-olds. It would also be very interesting to look at the impact of severity of disease and duration of disease on the future athletic performance. I'd just like to say a big thank you to Professor Proudman for his support with the study and my partner, Sophie Lanshoff. Thank you very much.
Thanks very much, firstly, for having me down this morning. So today, I'm going to talk a little bit about my intercalated degree research, and we're going to focus on the molecular characterization of canine Bcell lymphoma, and principally discuss a gene known as TRAF3. So TRAF3 is a gene which is involved in encoding a protein which regulates the non-canonical NF-kappa B signaling pathway. And this is known to be important in the pathogenesis of a subset of both human and canine B-cell lymphomas. So there's 3 aims of the study. The first was to validate findings from a previously conducted RNA-seq analysis. The second was to identify any novel variants in exon 9 of TRAF3, and the third was to predict the effect that these mutations might have at the level of the protein. So 60 canine DNA samples were screened using PCR and Sanger sequencing. 26 of these were from B-cell cases, and we had 9 matched controls from the same dog, or 9 matched controls from the same dogs, rather. 4 from T-cells with 1 matched control, and we had 20 from patients with non-malignant disease. So, all in all, 5 sequence variants were detected, and this is a selection of some of our data here. So the most common was a synonymous single nucleotide variant, and this was previously described in the RNA-seq study, and it was present in 31 samples, so from B-cell lymphomas, T-cell lymphomas, and from patients with non-malignant disease. The remaining 4 were all bona fide inactivating mutations, and they were found in B-cell lymphoma cases only. So the first is what we call a nonsense mutation. So that's this one here, and effectively what that means is that there's been a base substitution. So we've had a swap around, and we've incorporated a premature stop codon. The other 3 were known as indels, so a fancy word for a frameshift mutation effectively, where we've had insertion in these 2 cases of a 1 base pair and in the bottom of 8 base pairs. So what effect does this have at the level of the protein? Well, all 4 of these mutations were predicted to result in premature truncation of the TRAF3 protein, principally via loss of what we call the TRAF-C, or the math domain. So the math domain is the part of the protein which is important for its function. So, just a few concluding remarks. This was the first UK study to describe inactivating mutations in canine TRAF3 and suggest their role in the pathogenesis of canine B-cell lymphoma. We validated findings from a previously conducted RNA-seq analysis, and we frequently observed exon 9 of TRAF3 as a site of mutation. So this suggests that this is a mutation hotspot. But how is this important in a clinical context? Well, by furthering our knowledge of the molecular characteristics of the disease, this will allow us to subclassify the disease further, as they have done with humans, and in doing so, achieve 3 things. So number 1 is better predict the course of the disease. Number 2 is to tailor our pre-existing protocols better. And 3 is to develop new targeted drugs. So admittedly, with this study, we haven't cured the disease, but there is no doubt that by furthering our knowledge of TRAF3 and its implications in the pathogenesis of the disease, that we're now one step closer. So at that point, I'd just like to thank those involved and welcome any questions.
Hi, thank you all for the opportunity to be here and speak to you about my work today. So the data that I'm going to be presenting today comes from the Feline Healthy Aging Clinic. The clinic was established in early 2017 to support the cat prospective ageing and welfare study. To date, we've enrolled nearly 200 cats into the clinic, and so I'm going to be presenting to you some of the gait data today. The main question I'm looking at answering is what changes with feline gait as they age. So the material that we use, the method that we use, we have a tech scan walkway, an HR3, and we encourage the cats to walk across the pressure mat at their own pace. We use food rewards, we use lots of positive encouragement, and for some cats we provide a place for them to hide at the end of the pressure mat. For a cat to be successful to be included into the cohort that I'm presenting today, they have to have had no lameness on their initial orthopaedic assessment. They had to have crossed the pressure mat at a steady pace in a straight line, which you can see from the photos sometimes isn't very successful. And they have to have produced at least 2 successful walks for each time period that I wanted to compare. And those 2 walks had to be of a comparative velocity as well. So I had the results available from 7 cats at the time that I wrote this up. The sort of parameters that we were looking at, I did t-tests on the ones that were normally distributed and Wilcoxon tests on the ones that were parametric, the non-parametric data. So you can see for 2 of the cats, we had a reduction in the left fore swing time. And in one of those cats, the right fore also increased in the maximum pressure. Those 2 cats, there was no change at all on their orthopaedics findings at year 9. So I didn't detect any musculoskeletal disease in them. For 3 of the cats, we had no significant changes at all. For one of the cats, I had a reduction in the left hind stride length. And that cat did, on its year 10 exam, have a reduction in the range of motion in his hips. And for cat number 25, we had an increase in the force, impulse and maximum pressure on the left forefoot. And I also had a reduction in the impulse on the right the right forefoot as well. That cat, on his orthopaedic assessment at year 10, had reduction in the range of motion in his left shoulder, and he also had reduction in range of motion in his hips as well. In addition, we're also looking at pressure distribution in our cats as well, so the pressure across their feet. I'm doing something called a pediatric topological, no, that's not the right term, pedibariatric statistical parametric mapping, which is shortened to PSPM, which is how I always refer to it, so I had to practice the name quite a bit earlier. This is, to my knowledge, the first time this sort of type of assessment has been performed in domestic cats. I don't think it's even been performed in large cats that I've come across a paper on anyway. So it's a way of looking at statistical difference at a pixel level. So I've included the footprints from cat number 25 and cat number 2 and cat number 25. These were the cats that had changes in their maximum pressure over time. But you can see from these results that there's no statistical difference at all in how the pressure is distributed, although I'll see if I can get this to work. You can see in this one there is just a slight trend with the bluing at this end and the red at this end that the pressure is potentially slightly decreasing in the caudal part of the foot. And you can see from these 2 means here, you can see this part here is brighter than here, indicating the pressure is lower in this part of the foot at year 10. I also just wanted to include cat number 15 as well. So this cat had no statistical changes at all on its other measurements, but on the pressure distribution on its left hind foot, I did get a statistically different result. So you can see that where am I? There we go. There's a decrease in pressure in the caudal part of the foot, and you can see from the maps that your pressure peaks have really changed in year 10 to the cranial part of the foot compared to the caudal part of the foot. So that was an interesting finding in that one. I'm still not 100% sure what some of this means, and we do have a suspicion that the pressure mat that we're using potentially isn't sensitive enough for the lower pressures that cats use. We possibly need a more sophisticated or denser pixeled pressure mat. So we are also going to be doing a more sort of common four-quadrant approach as well to looking at the pressure distribution in these feet. So that work's currently ongoing. So really, in conclusion, I'm sort of hoping to sort of show that that gait changes in cats over time potentially might aid our early detection of orthopedic or musculoskeletal disease in cats as they age. There definitely are some issues with our study. We're not able to do, unfortunately, any diagnostic imaging for orthopedic disease due to cost and ethical constraints on the study. So it would be nice if we could, but unfortunately we don't have that information. One of the other things I am finding as well is that the cat's velocity does decrease over time. That may be a reduction in enthusiasm, but that also could reflect the development of musculoskeletal disease as well. I just wanted to give a quick thank you to all of my supervisors, Alex German, Gina Pinchbeck, but also particularly Carl Bates and Ethna Comerford, who have helped me with the gait stuff. And also a thank you to Jamie Gardner for all the Mat Lab assistants, Kelly Eyre for helping me cat herd, is probably the best term for it, and also to Royal Canin, who sponsor my studentship. Thank you very much.
Thank you so much to the RTVS for welcoming me here to talk to you all today about my research at the Royal Veterinary College. I'm a second-year PhD student there. I'm just talking to you about a slight sort of part of my PhD project, which I'm looking at doing some gene editing, which we all kind of hear about in the news, but I think hopefully I can explain to you a little bit more about it, so we'll go home a bit more clear about what that involves. So I'm particularly, at the RVC, we're particularly interested in hypertrophic cardiomyopathy, which is one of the most common heart diseases we see in cats. They get really massive left ventricles, you can see on the echo loop just up here. This is the muscle wall that's incredibly thickened. This is my cat over here, because he's very pretty, and he doesn't, well, I don't think he has this at the moment, but basically, when their hearts look like this, the cats die very quickly. They're quite light dying when they have this disease, and we can't really do a huge amount about it at the moment. So how do we help them? And basically, in cats, we know that in at least 2 breeds of cats, there's a gene mutation that causes hypertrophic cardiomyopathy, and it's a single letter change in the gene encoding myosin binding protein C, which is one of the proteins in the heart. And we don't really know why this gene mutation, or how this gene mutation goes from a single letter change to the disease that we see in these cats, which is obviously very severe. So I was particularly interested in this and looking at gene editing to kind of answer this question. And how does the gene editing work? So we use a technique called CRISPR, which was only developed sort of around 2013, so not very long, but it's an incredibly powerful tool for looking at new genetic diseases. And what it stands for is clustered, regularly interspaced, short palindromic repeats, which is why somebody who liked crisps decided to call it CRISPR. And basically what it means is that we can very specifically target genes in the genome and change basically whatever we like, anywhere we like, pretty much without many exceptions. And the CRISPR system is used by bacteria to destroy incoming viruses, but we kind of hijack the system to edit DNA in cells. The system incorporates 3 main parts. There's a Cas9 protein, guide RNA, and then a sequence that we introduce as well. And it's a bit like a guided missile in a way. Basically you have, well, that's how I like to think about it. It's not particularly explosive in the lab, obviously. But basically you have this Cas9 protein which sort of searches along the genome in your cells, searching for a place to attach and break the DNA. We then use a guide RNA, which we can specify the sequence. The sequence is then specific to a point in the DNA that we want to edit. We introduce all these into the cells. The Cas9 finds where it needs to bind, cuts the DNA at both strands, and then the cell gets very panicked and wants to repair the DNA. So it's at that point we hijack the process and introduce a piece of DNA, which then the cell hopefully then copies into place. And in that piece of DNA we're copying into the genome, we can put basically anything we like. We can introduce new genes, we can knock out genes, we can introduce single letter changes, and that's sort of why it's quite a relatively simple, easy process, and that's why it's become so popular. So in a human stem cell line, we've sort of recently done this, introducing the CAT mutation, which causes hypertrophic cardiomyopathy. And this was, I was actually in Spain when I received this data on holiday. I was meant to be on holiday, but I looked at the data anyway. So here, lots and lots of months of work. Basically, we managed to change here. This is a trace in the gene, GATC. And then this is supposed to be a C, but in the lab, we changed it to a T. So, yeah, quite a small thing, but for me, it was quite exciting. So then basically, you get a bit weird when you're doing a PhD. And then what we can do with these stem cells is turn them into heart cells. So here you can see some heart cells. This is sort of be about 400 times zoom, and you can see the cells are beating in the culture dish. So I spend most of my life looking down the microscope, looking at these cells. And what that means is now we can have basically unlimited supply of cardiomyocytes, so heart muscle cells, from these cells that have the genetic mutation introduced. And what we can now do is, sort of going forward, is looking at how these cells are different based on whether they have that mutation we've just introduced or not, and compare them in terms of how they're making their DNA, how they're making their protein, how they're behaving differently. And we can get lots and lots of cells to study this, and we don't have to kill lots of cats. The other things we can do is just basically I spend a lot of my time making pretty pictures. This is in green. This is myosin binding protein C, the gene that protein we're interested in. Actin is in red. Those of you with a very keen eyesight can see the striations in the muscle here and up here. And these are sort of the assays that we want to be looking at, sort of furthering understanding as to how this genetic mutation causes hypertrophic cardiomyopathy in cats, and really target down to see if there are new pathways that we can design drugs towards and actually try and help these animals to live a bit of a longer life. So thank you to my supervisors, David Connolly at the RVC, Jade here at the RVC as well, and Debbie Guest, who's my collaborator at my health trust, also to Bowring and to Win for funding the study. And I'm happy to take any questions.
So my name's Luke Gunter. Thanks very much for inviting me today. And I'm going to be talking about the effect of FST L3 deletion on trophoblast migration. So trophoblast migration is very important for key placentation. So you have your cytotrophoblasts, which migrate to the spiral arteries, and these help form them into larger bore conduits, so it's lower resistance for the blood to run through, so you have more efficient blood supply. You also have your syncytiotrophoblasts, which migrate to help form fusion layers to help facilitate nutrient exchange between mother and fetus. So in order to have successful trophoblast migration, you need key properties, and I've decided to focus on cell survival and actin polymerization in this case. So there's one very important gene called SIRT1, which is involved in deacetylating P53 and stopping the cell cycle arrest at G1 and G2 phases of the cell cycle. And in SIRT1 knockout mice, the placenta had very, very poor structure. Additionally, with regards to actin polymerization, FMNL2, another gene, knockout mice, showed decreased lamellipodia width and decreased density in actin nucleation. So leading on to FST L3, within these poorly vasculatured placentas, there's lots of disorders such as preeclampsia or poor intrauterine growth which occur. And in RNA-seq studies, they've shown that FST L3 is a key gene that's always upregulated or downregulated when these disorders happen. So how does FST L3 relate to SIRT1 and FMNL2 for the properties needed for trophoblast migration? And that's through this actually quite simple diagram up here. So what FST L3 is it stands for follistatin-like protein 3, and it's a functional antagonist of a molecule called activin, which is part of the TGF-beta superfamily. And what activin normally does is it binds to a type 2 receptor, which then recruits a type 1 receptor, and this phosphorylates the GS domain, which leads to phosphorylation of what are known as SMAD2 and 3, which then recruits SMAD4, and then this translocates to the nucleus. So once it translocates to the nucleus, it interacts with transcriptional factors and regulates downstream gene expression. So if in diseases such as preeclampsia, FST L3 looks like a key regulator, perhaps these transcriptional factors it's interacting with and the downstream gene regulation that's changing could be such things such as SIRT1 and FMNL2 involved in the properties for trophoblast migration. So to investigate this, I decided to do a scratch assay on BO trophoblast cell line. So we did this image taking every hour over 14 hours. So we did one with activin treatment, one with activin action inhibitor, which is SB and then the string of numbers, one combining the activin and the activin action inhibitor together, and also the control. So as you can see on the control, the black line, it's just very steady on the bottom there. Interestingly, when we used activin, represented by the pink line, there's a very steep initial increase in cell migration rate. And oddly, when using the activin action inhibitor, there's also an increase in cell migration rate over there, which is weird because it's exerting the opposite effect at the stimuli. And then interestingly again, when combining activin and the activin action inhibitor, it actually doesn't increase the rate as much as you would, because you might expect it to be synergistic, since both the activin and its inhibitor increase cell migration rate. So we wanted to find out why this might be. So what we did was we got some murine placenta FST L3 knockout and wild type, and we looked whether SIRT1 increased in FST L3 wild type or knockout, and also for FMNL2. And as you can see, in the wild type, SIRT1 and FMNL2 are much more bright compared to the knockout, so they're actually being down-regulated in the knockout. So in order to explain why perhaps the inhibitor and SIRT1 and FMNL2 were down-regulated in the FST L3 knockout, we came up with this diagram on the right here. So normally, as shown by the bold black lines, you would have activin, which would be causing SMAD2 and 3 to be phosphorylated, leading to transcription factor and gene regulation, increasing cell migration. However, when you use the inhibitor, perhaps when it's inhibiting activin, which is normally perhaps inhibiting unknown endogenous ligands, such as SIRT1 and FMNL2, which would make sense because they're down-regulated in the knockout of FST L3. This creates an override and increases cell migration via those pathways over there. In the future, we're hoping to do an FST L3 small interfering RNA transfection on these cells. So that way, instead of just using the activin action inhibitor, we won't be inhibiting other serine threonine kinase domains, and we can see FST L3's action in vivo that way. Thanks very much for listening.
Hello, I'm Charlotte and I like turtles. So I'm here to talk about comparisons of dye-binding methods for the determination of albumin in loggerhead turtle plasma. Now the reason for this is turtles present, well, reptiles in general present with very few clinical signs when they're ill. A very ill turtle can look very much like a dead turtle, and it can look that way a long time before it becomes a dead turtle or decides to be a live turtle again. And in a rescue and rehabilitation centre where you have a limited number of resources, how long do you keep a turtle in a tank before you give up and think about euthanizing and letting somebody else have that tank? Because they can hang around for like 18 months. So at the moment, these sort of decisions are made on non-objective indicators. So plastron concavity, epibiont coverage, is it moving at all? And these are not quantifiable, and these are not scientific. So I was interested in looking at clinical pathological indicators that are objective and cost-effective to try and work out which of these turtles were going to make it and which ones weren't. Now, from previous work by Stacey et al. In 2018, albumin has been found to be a promising non-prognostic indicator. However, there is not a reliable assay for albumin in reptiles. Well, there's protein plasma electrophoresis, but that is expensive, and if you're thinking of taking serial albumins and you're looking in a rehab position, this is not possible. We need something cheaper. Now, the common method clinically to measure albumin in mammals, bromocresol green, dye binding, is known to hugely overestimate in most reptile species studied, particularly chelonia. And this is because it binds not only with albumin, but with alpha-2 globulin and gamma globulins. Bromocresol purple, which is a similar phthalate dye, also binds with prealbumin, but does not bind with the globulins, and therefore is possibly going to be more accurate for albumin determination in reptile species, but it has yet to be assessed. So I blood sampled 21 adult loggerhead turtles at Decamere Research and Rehabilitation Facility in southeast Turkey. And we found that in the 21 turtles we sampled, none of the albumin levels were identical when measured by the two methods. And we used commercial kits by Roche. The average difference between them was 9.6 grams per litre, with bromocresol green overestimating, well, being higher consistently. When we use passing Bablock analysis, we see that we have sorry, this is stats, I don't like stats. It's proportional bias getting higher as the concentration increases. Although the bias was, when you look percentage-wise, the bias was higher at the lower values of albumin. And that's the Bland-Altman plots. Now, unfortunately, the main limitation of this study is that we were unable to get plasma protein electrophoresis run on the 21 samples in time for it to be included in my master's thesis. However, this is hopefully happening in the next couple of weeks. I'm going back to Turkey on Monday next week to try and get it run, because of CITES regulations and Turkish law, we cannot get the loggerhead plasma samples out of Turkey, and it's actually much, much harder to get a plasma protein or electrophoresis run in Turkey than you think it would be. So, once we have that value, we should be able to try and validate whether BCP is underestimating or BCG is overestimating, or whether it's actually a combination of the both, which is what I suspect. We also took total protein by refractometry on all the samples, and the BCP correlated more closely to that, whereas the BCG was kind of all over the place. The BCG values were not consistent with anything. So I'm hopeful that BCP will be a more reliable indicator for albumin in reptile species going forward, and then we can sort of extend the study into other species of reptile. And I'd like to thank Professor Jakub Kaszka of Decamere and Dr. Mike Waters at the RVC, who are my project supervisors.
The transition into veterinary education is a hugely significant time for students. Not only do they have to adjust to new academic surroundings, but they also have to adjust to new social surroundings, like moving away from home for the first time and dealing with new financial pressures or losing their kind of social support networks. Previous research has looked into very specific groups of how they move into veterinary education. So, for example, international students or postgraduates and mature students. But I wanted to take a little more of a kind of wider frame look at how students progress into veterinary education. So I did this as part of an intercalated postgraduate certificate in veterinary education at the University of Nottingham, where I'm finishing my last couple of years of my undergraduate. So a little bit about what I did. So I did a mixed method study. So we wanted to collect quantitative and qualitative data, and we wanted them to feed into each other. So first of all, I set up a discussion forum. This was done on Facebook, and we had a small online private group, which we invited some students at the end of their first year to join, and we asked them questions on a regular basis, I think it was every day for about a week, on how they found the transition into vet school and how they found their experience, and how did their experience match to their expectations beforehand. We then thematically analysed the results, and then we used the findings of this to help put towards an online questionnaire that we then rolled out to the first year students when they arrived in September. We then got some data from the survey, and that we analysed that quantitatively. But however, the survey was very specific to the Nottingham course, so what I'm going to talk a bit about today is the more discussion forum and qualitative analysis. So using the online discussion forum on Facebook was a really novel way of collecting data. It had never been done at Nottingham before, and it was kind of interesting to see how it went. So we managed to get some really good data, and students were really engaged with the group. So I'd post questions on there, and they would be able to comment their replies. And what was really nice to see was they actually interacted with each other, and they'd comment on each other's comments and kind of have an actual good discussion. So that was really nice to see. So from my thematic analysis, I gained three main themes. surprise and uncertainty, independence and responsibility, and work-life balance. So students frequently talked about their surprise when they came to university. They talked about how they were surprised about various different aspects of the curriculum. So for example, practical teaching, clinical relevance, and the level of independent study required. They also talked a bit about their surprise when it came to how demanding the course was. They all expected it to be a kind of high demanding course, but they were also kind of a bit overwhelmed with the volume of work and the amount of content they were expected to learn. When it came to uncertainty, many students talked a lot about how when it came to university, they didn't have a specification or kind of the guidelines that they were so used to. From coming a very school-based education, which is very assessment-driven, where they've got their specification of this is what you need to learn to pass your exams, actually coming to vet school where there isn't that, and it's kind of a much more wider frame of you kind of can learn as much or as little as you want to learn as long as you kind of fit near learning outcomes. It was kind of a bit different for them to get their head around. And they often felt ill-prepared for this kind of adult-based pedagogies, so they weren't kind of really sure of how to deal with independent learning. And they felt kind of a bit unfamiliar with this new increased responsibility. A lot of them kind of did struggle with the independent learning, and many suggested that they weren't, it wasn't something that we thought was going to happen. They thought they'd be having a bit more of a regular kind of check-in. So students often said actually they were expecting homework when they came to university, that they thought they'd kind of regularly get contact with lecturers being like, oh, how are you getting on with this? Are you engaging and kind of getting through the content? And finally, one of the themes that kind of was quite surprising for me, because when I went into this, I kind of looked at it from an educational point of view, but actually what came out a lot was work-life balance, of students talking about either how they were struggling with the course and how they were finding some areas quite difficult, but actually they also felt really supported, and how they felt that the nice student community that had been built was really kind of good and strong, and that they felt that it was, they were able to engage with other students in a nice way and kind of felt supported. So I thought I'd move on to a few quotes that I got. So these really emphasize some of my findings. So this first quote emphasizes that actually there is quite a disconnect between school-based education and veterinary education. Students are really driven by that assessment-based learning at school of kind of, I remember when I was in A-levels personally, it was very much of, if the question says this, put this and you'll get the answer. If the question says this, put this one word and you'll get the marks. Whereas actually at veterinary education, that's not what it's about. It's all about kind of encouraging analysis and critical thinking and a bit more reflection and evaluation on your answers, and actually thinking how to adapt and adjust those answers to things like the individual, the scenario, and the patient. And actually, to have a first-year student to kind of think this, I was quite surprised by the end that they'd realised that by the end of just first year that was what they were supposed to be. And then my second quote kind of reiterates a bit more about the kind of work-life balance and kind of student support, that actually it is a kind of a hard course, and they are expected to be hard, but also they're not quite ready and kind of not quite adjusted to dealing with all of these extra pressures. A lot of students talked about how they moved away from kind of their close support networks of family and friends at home, and they're kind of thrown in at the deep end where now at university they've got no one and they've got to kind of figure it out for themselves. But it was really nice to hear again in the results that students actually bounced off each other a lot, and we have a lot of programs at Nottingham, such as like a big vet, little vet, where students really felt supported by that. So where do I see this kind of going in the future? So I think, to conclude, there is a clear misalignment between school-based education and veterinary education, and actually that maybe future projects do need to be enlisted that kind of offer a collaborative between schools and veterinary to kind of help align that better and kind of smoothen out that transition. That is everything for me, and I'd also like to thank my supervisors, Dr. Kate Cobb and Dr. Erica Gummery, for all their support, and also Professor Liz Mossop, who's here, who gave me the intercalation post to start with. So thank you.
Thank you. I'm going to try and do something a little bit different now. So I want to use everybody's imagination here, and I'm going to transport us to Croatia. And you've all had pretty great careers, but now we're veterinary public health officials working for the government. Now it's Friday afternoon, you've had a pretty good week, you're enjoying a coffee and you're sitting scrolling through Twitter. And that's when you see a photo. And it's a photo tweeted by the Croatian Ornithological Society, and it's what looks like to be a dead mute swan. Now, you look at the location tag, and it's the Crna Malacca Ornithological Reserve. And you recognize this place because it's previously assessed as a high-risk zone for high-path avian influenza outbreaks. And then you look at the hashtag, incoming avian influenza outbreak. And I mean, this is your job and you haven't heard about it yet. What's going on here? So you finish your coffee and you go to your desk and you start thinking about where this came from. And that's when the phone rings. And it's the president of the Croatian poultry industry. And they ask you if you've heard about the mute swan. And they want to make it very clear that they don't want a repeat of 2005, 2006. Because in 2005, 2006, there was 9 isolates of H5N1 across Croatia, only in wild birds. No spillover to poultry, but consumer habits dropped by 20% in poultry consumption. They took a big hit. They want to know what you're going to be doing about this potential outbreak. So you get on your email and you think, maybe I missed something, maybe it got sent to the junk mail. And that's when you see an email from one of your veterinary public health colleagues from Hungary. And they're asking what's going on with this outbreak. They want to know what you're doing because they don't want a repeat of 2016, 2017. Now, for those not familiar, that's when there was an epidemic of high-path avian influenza across Europe. There were 22 outbreaks in Croatia, 11 in wild birds and 11 in poultry, tons of mortalities of wild birds, and a lot of culling of backyard poultry farms. You realize you need to act fast. What is going on? Now, this is where my research can really improve your day. So I've developed a mathematical model using real-time migration dates to analyze how high-path avian influenza outbreaks create or result in mortality in wild birds and backyard poultry. More specifically, I'm interested in the overlap migration of wild bird species. So I've got 4 different populations in the model. I've got mute swans. And the reason I chose mute swans is because they're a sentinel species for high-path avian influenza. And what I mean by that is they all die. When a mute swan is infected with high-path avian influenza, it has 100% mortality, and they have always been the first species detected in Croatia in previous outbreaks. And that's because they're quite big. I've also chosen 2 populations of mallards, a migratory and resident population. And mallards are known to be asymptomatic carriers of high-path avian influenza. So they get the disease, and they've been thought to be spreading it from places like Asia, where it's endemic, over to Europe. And then finally, I have backyard poultry. And culturally in Croatia, it's quite common to have poultry in your backyard. And because they're free-ranging, they're at a high risk for avian influenza outbreaks. So using these real-time migration dates, I can create risk assessment model predictions. So I've run my model 100,000 times, and it's giving us an output of the number of mortalities based on the different parameter values. We can then use these risk assessments to implement risk-based control strategies. And what I mean by that is, if we plugged in the migration dates from this year, and we saw that it was a high-risk year, we could have said, okay, everybody with backyard poultry, keep your birds inside. Or, if we saw it was a high-risk year, we could have changed our passive surveillance to an active surveillance and been out in the field and detected this early. Now, with avian influenza, we don't know what the next strain is going to be, and we don't know who's going to bring it in. There's also a lot of changes with the climate, and that's causing changes in migratory bird behavior. These birds are going to be migrating at different times and are going to be overlapping at different locations. So you're sitting at your desk, you're stressed out, your beautiful weekend with the family is now interrupted. If we ran my model beforehand at the start of the year, we could have predicted that this was a high-risk year for avian influenza. And doing that, that call from the president of the poultry industry would have been thanking you for the precautionary measures that you put into place. And the Hungarian veterinary public health official, again, would be thanking you for your global reputation and ensuring that this is not spread across Europe. Thank you.